This is TWIS. This Week in Science, episode number 958, recorded on Wednesday, January 31st, 2024. Why is the ocean venting? It's not like Justin, really. I'm Dr. Kiki, and tonight on the show, we will fill your heads with muscle bots, mouse squeaks, and lost city vents. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience. Disclaimer, disclaimer, disclaimer! I disclaim before I proclaim, this show is not the same, even by name, as we try to explain in another frame and may go against the grain, not seeking fame, never in shame, food for your brain. So let's all exclaim, it's This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I wanna learn everything. I wanna fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I wanna know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Hello, happy day of science, everyone. Welcome to another episode of This Week in Science. Tonight, we do not have a Justin, we do not have a Blair, but we do have a guest who I will introduce to you in just one moment. Thank you so much for joining us. I see you out there. I'm so glad you're here for another week of science. I have a bunch of science news stories involving genomes and muscle bots and mouse squeaks. And I would love to introduce you to our guest tonight, Sarah Treadwell, a science communicator who focuses on astrobiology, but is well-versed in many other things. Sarah, thank you so much for joining us tonight.
Thank you for having me. This is so exciting.
Honestly, I have to say, big thanks to Graham Lau from Ask an Astrobiologist, the podcast, because I reached out today realizing that I had no other co-hosts for the show, and it was a scary moment going, where do I go? And I reached out and Graham was unavailable, but he was able to connect me to Sarah. So, and thank you. This is last minute and amazing. So thank you for being here.
Yes, thank you, Graham.
Thank you, Sarah. So we'll get into a lot more discussion with Sarah into the show about what makes her a space case, what got her interested in astrobiology. But first, as always, we're going to dive into a few news stories and talk about what is This Week in Science. And as we jump into the show right now, I want to remind all of you that if you have not yet subscribed to This Week in Science, you can find us broadcasting, live streaming, weekly, Wednesdays, 8 p.m. Pacific time on YouTube, Twitch, and Facebook. All those videos are there. So if you are subscribed and hit the little notification, you can catch us live or watch us whenever you want. We're also a podcast all over the podcastoverse, and just look for This Week in Science. If you are curious about other aspects of the show or just can't remember all of this information, head to twis.org, and that's where you can find all these links and all this stuff. But that's the spiel. Now it's time for the science. Okay, diving in. My first story for the evening tonight has to do with mapping genomes. And we've been talking about mapping genomes for a very long time on this program. A long time ago, the Human Genome Project, it took 13 years to map the entire human genome. And that wasn't even the most accurate map of the human genome because of the methodologies that they used. And we've been mapping other organisms over the decades since this technology of sequencing and splitting up the genome into little tiny puzzle pieces that then have to be fit together in just the right way could be done the most efficiently. It's kind of like going from like the little 2-year-old puzzles where it's the big blocks to like the really complex 1,000-piece puzzles that have tiny little pieces and a lot of just one color. This is where we are now. And Researchers who just published in Nature Biotechnology, they published their genomes, which are the big news, is more accurate and they were more efficient. So instead of years or months to be able to get these genomes, they were able to get the genomes in days. I hear a puppy dog. That's good.
His name is Hubble, by the way. I need to refrain from going Hubble.
Uh, no, if you just have to, bring Hubble in, for a little love,
Maybe later.
we won't complain. So the exciting part of this assembly is this new pipeline that they've developed. And so they published just this last week in Nature Biotechnology, and the paper is Scalable, Accessible, Reproducible Reference Genome Assembly and Evaluation in Galaxy. What's Galaxy? Galaxy is an open access, open source software that is available to researchers and people in the public online. It's part of the Vertebrate Genomes Project, which has been running for a good long time. And they are trying to map 70,000 genomes. Like, that's the goal, is to map 70,000 species genomes for the vertebrate species on the planet. They're using the Galaxy infrastructure and the public instances to allow the collaboration. This allowed them to compare the genomes that this new publication was developing against what had already been assembled. They started with the zebrafinch, which is my favorite little tiny bird. I spent a lot of time with Taenia guttata in my graduate years, and it's one of the many vertebrate species that have been previously sequenced. But with this assembly and this new research, the researchers involved in this are able to actually make available the software, the new algorithms, and the new databases that are hopefully going to enable what will one day be an understanding of the multitude of genomes, vertebrate species specifically, that are present on Earth, and be able to actually compare all of them in a much more accurate way. And the big issue with this study is that it needed to be scalable. With the time it takes to sequence, or it has taken to sequence, vertebrate genomes historically, the idea of getting to 70,000 would take decades. I mean,
Wow.
centuries.
Yeah.
Yeah.
Yeah, I was gonna say this is definitely the kind of the way that things are moving with data repositories and being open source and having this accessibility for researchers to be able to compare and replicate. I just was in Santa Barbara doing some training on R coding for Arctic data repositories.
Oh,
So
cool.
this is so in line with that same sort of thing.
I mean, the microbial life and the other. I mean, this is stuff that we know is there, and we can see. Look, it's a bird in a tree. Let's take a feather. Let's take a skin cell sample and see what's in it. But like so much of the Arctic stuff and the stuff in the water and the sequencing of the unknown, it's like, what is there?
Yeah.
And it's real investigative digging. But this is exciting because it hopefully will actually allow this kind of sequencing to scale and move forward in a way that will actually get us to lots more genome sequences in less time and more accurately. They're using also the what is the name of the group that I just blanked on, the company that is I'm forgetting the name of the company, but it is a very it's, it's one that is very well known in the space of chopping up genome sequences and being able to put them back together. And I don't know why I'm not remembering that right now. Anyway, little tiny puzzle pieces, and that's what they're using. Should we move forward into robots?
I'm all for robots.
As opposed to my lack of memory.
No,
Um,
no, you're
yeah,
fine.
it's all good. It's okay. Um, how do you feel, Sarah, about robots in general? The idea of robotics and how they work.
This is a you're asking the right person. I actually own a small robot. Her name is Sagan, and it's a following robot that uses some AI technology to track the person that it's programmed to follow in that moment, and it
Mm.
maintains a certain distance, and it can carry up to 20 pounds, and it's on two wheels, and you yeah, just use
Did
it.
you steal this robot from NASA?
No, I did not. I did. No, admittedly saw an ad for it on TikTok, and I was like, "Ooh, do I need a robot? No, but do I want this robot? Yes."
Yes.
So it's called a Jita bot. So yes, I'm a big fan. I'm a big fan of robots.
Well, in space exploration, we know robots are going to do the work for a while, for a while in other places where humans cannot be. They're going
Mhm.
to collect samples. They're already on the surface of Mars. We've got robots going out all over the place. But one of the questions is, how do we make robots more efficient? And how do we, right now, it takes big batteries. A lot of them have to be plugged in. Uh, the stuff that we're looking at the really fancy robot companies where they show their robots doing dances and backflips and stuff, they're
We know
not,
who they are. Yes.
yeah, they're programmed and they're only working for short periods of time. These are not bots that are just like, hey, I can hang out for a whole day and then, go to sleep. But what if robots had skeletal muscle? What if they were able to function based off of the same biology that vertebrates, mammals, humans use?
Yeah, the be. I mean, are you? This one, I guess I didn't read farther along in this paper because I read all of these ones that you sent. So, like, are we talking like 3D printing, human kind of parts and putting it into a robot? Is that kind of the proposal that's moving here?
No, this is not 3D printing right now. It is culturing skeletal muscles and creating actual muscle cells that work in the same way that, say, a group of muscles working together in a myocyte working with other muscles in a muscle bundle to be part of a larger muscle organ, to move a limb. And these researchers have this is not taking over the world yet, everybody. I just letting you know. Right
Yeah.
now, they've got these little teeny tiny robot legs made out of plastic pieces that were probably 3D printed and with muscle cells connecting the tops and the bottoms. And they have been able to energize those muscle cells with electrodes and alternate the on, off, on, off, and create a structure which is like walking.
Yeah. So this
And
is like Frankenstein, but real life.
yeah, but only if these little robot legs are in a cellular nutrient bath. Like they can't come out of water because the muscles dry out and then they shrink because the cells are dying. Yeah.
Interesting. Yeah. So you're wondering, how do I feel about that? Like what? Like how does?
But
Yeah. It's
where this will go eventually is definitely further than where it is now. But
oh, for sure.
yeah.
Yeah. Yeah, I don't know. That it's a I think this is going to be, at least from a science communicator perspective, this might be a hard sell for a while. I'm not going to lie. Like I think this might freak some people out. And by some I mean a lot of people out. Yeah,
Yeah.
I don't know.
Yeah, it's a very simple design, and it's right now they're sticking electrodes into the water to enable the movement. I'm going to share this from the journal Matter, where it's published. Um, we've got these little electrodes in a nutrient water bath, and when they turn on or off, the different legs move. It's not beautiful movement. It's not a ballet dancer. It's not, but it is the beginning of possibly leading to lighter weight robots that are made of maybe more efficient materials. We know biology works very well. So, can this turn into something more? Will they ever be able to get past this need to have the robot muscles in water or a nutrient bath? Where is it going to go from here? But it's an interesting start. 3D cultured muscles for robots. Dun, dun, dun. I'm a fan. I think robots always deserve the benefit of the doubt. It's the programmers that are maybe more problematic.
Yes, I would agree.
Or the data sets.
That's an interesting twist, though. So, and I have a whole train of thought going on that, but there are some other stories. So
we do have more stories. Yes. Eric Knapp in the chat is saying, wasn't there a documentary on this kind of thing called Blade Runner? Yeah, we saw how that worked out. Hopefully science fiction will help us move into the future better. All right, everybody, if you've been out at night after the sun goes down, the lights come on outside, and you've probably seen clouds of insects, moths, gnats, other nighttime insects come and gather and seem kind of chaotic, flying around those lights. And everybody says, oh, it's just moth to a flame, right? They're just attracted by light. But why are they attracted by the light, and why do they fly in the way that they do around those lights? Some researchers just publishing in Nature Communications, they have published their work this last week, which I find fascinating. A researcher was like, oh, okay, why do they fly around the lights the way that they do? Why doesn't it seem like normal flight? This is a weird behavior. And they put a moth to a light in the laboratory and realized that the moth was reorienting, so its back was to the light. They're not coming up and facing the light like as if they're, worshiping it. They come to the light, but then reorient, and their back goes to the light. And so they started doing a lot of really high resolution, three-dimensional space models. They had, the researcher says, I let a large yellow underwing moth take off from my hand and fly directly over a UV bulb, and immediately it flipped upside down. They didn't know if they would be able to see this in the wild or not. And so they went to Costa Rica and did a whole bunch of experiments with lights and saw what the insects did, and were able to determine that this is normal behavior, that the flipping around is what the night insects do, because they think the lights are the brightest thing in the sky, which would be the moon or the stars normally. But because we have artificial lights, suddenly we have created this new bright source. But then they also are trained by gravity. So the gravity of the Earth tells them how to orient. And instead of orienting to the moon or the stars with their back to that, they are now orienting and trying to orient to this artificial light source. And in looking at the light source and the data that they got from it, they were able to actually model why and how, and be able to predict the movement of the moths, and that it's not normal flight. It is specifically within this controlled environment. It's chaotic, and it only occurs as a result of the artificial light and the combination of the gravity. But then they did this really neat addition to it because of reflected light, right? So they were like, oh, what if we have a light shining at the ground, and what's going to happen to the insect then? Where is it going to go? And they found that they crash on the ground. When the
Huh.
light is shining at the ground becomes the reflective source, and the insects get confused and they crash on the ground. But if the light is facing up, they are more likely to fly upward, right side up, and normally.
This is
So
like the
depending.
equivalent of the cat, like the memes with the piece of bread on the back, and then it doesn't know which way to fly.
Right. If you put
Right?
buttered bread on the back of a cat, where it's just going to hover forever. Yeah, never falling.
Yeah, no, that's interesting though that the gravity is something that they orient themselves with because we know that a lot of species use the magnetic properties of the earth to know how to migrate and stuff like that. So I guess that completely makes sense to me. And as an astronomer, I would definitely say it's very frustrating. Oh my god, I'm gonna have to take care of this dog in a second. To
Hubble, we
have
love you.
to have these light sources, because it does, it not only messes up with your astronomy, but it messes up with these animals and these, different, the migrations and the bugs and that kind of stuff. So yeah, interesting study.
Yeah, and what I wonder as they move forward with this research, how will this help us with our urban environments? How can it help us do new lighting design so that we aren't interfering as much? So right now we have street lights that come down to help
Mhm.
us see our sidewalks, but what if the lights were shining from the ground up instead? How would that change the way that things worked?
Yeah, I don't know.
I don't know either.
I don't know.
Yeah.
Yeah, it's interesting, and I don't know. Pretty I mean, couldn't complain like studying a bunch of moths in Costa Rica. That sounds like a pretty fun job.
It sounds like a fantastic job, but who knew that these nighttime insects decide to fly up to the light and then turn their backs on it?
And then flip on it. Yeah.
And then that makes them act stranger than normal. Um, Speaking of, no, not stranger than normal, mice. I like mice. Anyway, terrible segue. Uh, Mice communicate in many different ways. We've talked over the years about not just alarm calls or squeaks related to pain, but mice also have ultrasonic sounds that are very, very high pitched that allow them to communicate for mating and for other social communications. There are even some mice that we know sing, and there are rats that we know to laugh. So there are all sorts of vocalizations that take place in the mouse world. Researchers wanted to know if all of the same neurons in the brain were responsible for all of these vocalizations. So basically, across many species, we know that there's an area of mammals called the midbrain periaqueductal gray region, and it regulates vocal production. And if you artificially stimulate it, then it causes a squeak in a mouse, or it causes a person to go, ah, or it makes vocalizations in many different species. But how they're organized and all the different types of neurons that come through that periaqueductal gray region isn't very well known. And the researchers were like, I wonder. So they looked at a bunch of mice and they blocked neurons in this periaqueductal gray region, the PAG region. And they determined that if they blocked these very specific when they blocked these neurons, it blocked the high ultrasonic calls, but it did not block the squeaks of pain or the alarm calls. So it's the real communication between the mice that gets blocked when those neurons are turned off, as opposed to like the kind of alarm or pain responses. And there is this amazing video that I'm going to try and share it with sound. So I hope it works out. And in it, the researchers. Oh, did I share it with sound? I have to wait. Stop sharing. Hold on. I have to make sure I've got all of the all of the things also share tab audio. Yes, good. Okay, good.
These are those technical difficulties.
The technical things that we want to make sure that we're working through. Yes. So in this video, the researchers have a male interacting with a female before and then after these neurons have been turned off. And in the beginning, can you hear the high-pitched squeaking?
Yeah.
So in the beginning, these ultrasonic calls are the male calling to the female. And then the male and the female keep interacting. But after the neurons are turned off, the male is no longer making any sounds that can be detected. Yeah.
But they do still make the ouch, noises, right? Because from an evolutionary perspective, that kind of makes sense, right? That's probably coming from a more survival part of your brain that's, that reflexive kind of response, but interesting. And like also the fact that we're like, yeah, and we turn off this part of the neurons. Like, what? You know what I mean? Like, what? That's just so wild. That's just
it's amazing that they're able to specifically affect just certain groups of neurons. But they were able to also extend this to females. And so there's activities, the behavior still happens, but they're just silent when that area of the brain is turned off. And now it's, what does this mean for how brains are set up in social animals, the communication that takes place, and how the brain works to allow vocalizations at certain, specific times or not.
Yeah.
So, yeah. But eventually, I like we're just trying to figure it out for how it works in the mice. But of course, there's always maybe if we understand this in mice, we can look at how this affects and it might apply to humans as well.
Yeah, sure.
Yeah, down the line. And moving on from mice, let's talk about enzymes and plastics. There's a lot of them.
Yeah.
You go to
yeah.
The single-use plastics are the ones that are really problematic right now. Bioplastic polylactic acid, PLA. It's very often from a natural source like soybeans or corn or potatoes even. And very often it's like, great, it's good plastic. Well, it still doesn't break down easily like it's a polymer. That's what it is. They took a natural source and turned it into the polymers that make up plastics. So researchers publishing in Cell Reports Physical Science from King's College London, they were looking at this issue of, you go to the coffee shop, you get a cup of coffee, the inside of it is lined with the PLA so that the coffee doesn't go through the paper of the cup and you can enjoy your coffee. Then you go throw it away. It goes in a landfill. It takes a long time to break down. We know that there are microbes and there are enzymes that could possibly help with this, but what if we could develop processes to use enzymes that are already in use in our industrial processing or in our manufacturing system? And in this particular study, they have described a general route to retooling hydrolytic enzymes toward plastic degradation. And they used one specific enzyme that is lipase B, and it's from a Candida antarctica species. Yes, but it's used in laundry detergent. So you have this enzyme being used on large scale very regularly. And in this study and this process, what they show is success in being able to get the enzyme very easily to a state where it will grab onto that PLA and tear the polymer apart, right? It'll take the plastic pieces and rip them into the organic bases that they come from. And they show full degradation in 24 hours and conversion to monomers instead of the polymers, which are long chains, within 40 hours at just 90 degrees Celsius. So this is something that is within reason for industry, for our recycling, for the potential to be able to grab these things before they end up in a landfill or even as they go through a landfill and use these enzymes to to make plastic recycling better.
More efficient. Yeah.
More, yeah.
Yeah. Yeah.
So
And
we put less in the ocean.
and your laundry's really clean. No, that was one of my favorite things learning in biology was about how microbes and enzymes break down things. And that's why now, think of even just when I was a kid, you had these washing machines and you put your hot water in there and now it's like minimal water and cool water and you get clean laundry and it's because of enzymes.
Enzymes are just they are really they're what allow cells to function. They are really helping us move forward, and I mean, I'm excited about how artificial intelligence, these learning algorithms are going to enable us to identify more and better enzymes that will potentially, or even enzymes that we already have in use that we can use to even, better living through chemistry.
Yes.
Make our lives better.
Science.
Yeah, I don't know. I really do hope that we can figure out the plastic issue, because that is one of the big ones moving forward.
Yeah, I mean, that's a layered problem too, because when you're thinking about these biodegradable plastics, right, that come from corn or soybeans or stuff like that, there's also a problem in that too, that these huge scale productions of these crops to make these products and those monocultures are not super great for the earth either. So it's, it's to me, whatever I look at an issue, it's never simple. It's never black and white. There's no like, here's the problem and I'm just going to pull that problem out. It's always very nuanced and it's a scale. So yeah.
Yeah. And I think that's one of the big problems and needs when it comes to science communication is getting past, a history of teaching students that science is right or wrong answers, and actually educating people as to the nuances and the complexity of all of these issues and the systems that are at work on our planet, within our society, that make things work the way that they do. So yeah,
absolutely.
It's never black or white. No. I mean, I'm watching For All Mankind now, so yeah.
Oh, okay. I really haven't actually gotten into that much. So yeah, I mean, I know I should, but
there's no shoulds. It's just if you do or not. Do or do not.
Do or do not.
Yes. It's a very Yoda type saying. But anyway, okay, we have hit the first half of this show, and I just want you all to know that this is This Week in Science. We are running through the stories, and I'm so glad that you are here with us right now. If you enjoy TWIS, please share with a friend today. Help somebody else to subscribe. Let them know where we are on all of the different platforms where we stream live and where we podcast. Also, if you really love the show and want to help us continue, head over to twis.org and click on the Patreon link, where you can join the Patreon community of supporters for TWIS, and you can choose your level of support per month, $10 and more, and we will thank you by name at the end of the show. There's also our Zazzle store, so you can hit the Zazzle link and take a look at some of our TWIS merchandise. Many items have been created by hand, art created by Blair. So if you like science art in her Blair's Animal Corner segment, there's a calendar available. I mean, I know we're at the end of January, but there's still 11 more months to go. You have time. And there are many other things out there. But really, we thank you for your support. Thank you for being here. We really can't do it without you. And we're coming on back with more This Week in Science, and it's time to reintroduce our guest for the evening. Sarah Treadwell is a science communicator who currently is developing a book and a planetarium show about the Lost City Hydrothermal Field. What is this? We need to know more about this. She's also working with the Blue Marble Space Institute of Science and the Ask an Astrobiologist podcast, and pursuing a PhD in communications right now. Sarah, thank you so much for joining us this evening.
Thank you for having me. And
You're
actually, I haven't
welcome.
really talked to many people about the book thing. Not that it's a secret, but I just haven't. It's been a little like kind of low key. So you heard it here first.
I saw it on a NASA bio, so
Oh, okay.
it's
Oh, then I must
it's
have,
on
yeah, it
the
must have been
internet.
somewhere. It must be my scope project, I think, probably. Yeah.
Yeah.
Um, yeah, it's not a secret. It's just I haven't really like blasted it out there yet because it's still in the works. But yeah, no, thank you for having me.
So, the planetarium show you mentioned is the Scope, and that is funded by a NASA Scope grant. Is
Yeah.
that's what's going on there? What is happening
Yeah.
with that project?
So the NASA Scope grants are C grants for connecting subject matter experts to the SciAC teams, which is an initiative by NASA to kind of broaden science communication, I guess, in the best way to explain it. There's a lot of initiatives within the SciAx teams, but the SciAx teams are different projects that NASA funds. And so I am partnering with Open Space, which is an open source kind of data repository. And what they do is they are basically building the universe and
What?
and they're doing it to scale. So it's like a software that you can zip around the universe and everything is to scale. And so when I tell people you can easily get lost in open space, this is not an understatement because if you know anything about space, it is really, really big. And so
Understatement. Yes.
yeah. And so what they do to try to help people, because it's very easy to get lost, is they build these guides. And so say you're like, I really want to learn about all of the research and the missions we've ever done to Venus. There's probably a guide written for that. And so it will like guide you through the open space program. So it's not you trying to figure out how to fly there on your own. And that's what they call it when you're moving around it. It's flying. And so I was sitting in a meeting and Open Space was presenting and they were talking about a program guide that they had developed for astrobiology. And then they went on to say, yeah, but the only thing we don't have is going to hydrothermal fields under the water on Earth. And I was like, oh, darn, because I just sailed on a ship for 2 months that went over a hydrothermal field. And that would be really kind of useful for me as a science communicator. And so then I had someone in my comments going, oh, this is what a scope grant is for. And so that's kind of how that all came about. And so it's super exciting.
That's
Not only are we
amazing.
going to put the vent field in this software, but we're also going to build the ship in there as well to really connect the concept that it's while it is, you know, a lot of NASA missions built in there, there's still so many scientific organizations that make the magic happen. It's not isolated in a vacuum. And so you often find these big kind of things have a lot of funding from different countries, from different scientific organizations. So I really wanted to bring that to my guide that I'm writing, that, you know, it's not an isolated thing to, you know, have astrobiology research. It takes everybody. And that's what I like about astrobiology is that it's not just biologists. Actually, there's even a joke within it that it's very few biology people. It's a lot of like geologists and chemists and planetary scientists and, you know, lots of people make that happen.
Yeah, there's a lot of chemistry and physics before you actually get to life.
Yeah.
And yeah, there's a lot of stuff that underlies all of it. So yeah,
Yes.
how do you get to the understanding of what could make life possible before that, you know,
Right.
the chaos of ecosystems starts to
Exactly.
evolve?
Yeah. Which leads me to, you asked me, what is the lost city? And that's where, you know, I'm really
Yeah.
excited to work on this project because I didn't set out to be the kind of expert, if you will, on the lost city by any means, but it has started to consume my life. And now I know this place very, very intimately. And to your point, basically the lost city is a place where many scientists think chemistry might have turned to biology back in history, not this vent field, but this might be an analog to a system that perhaps this is where life may have gotten started because there are chemical reactions happening in this area that produce hydrogen and hydrocarbons. And so it's very feasible that this could be the place where maybe life got its foothold on Earth. And so that connection to space is that also many scientists think that therefore, if this could be happening on Earth, it could be happening on icy moons of like Jupiter and Saturn.
So this hydrothermal field, we've talked about hydrothermal vents on the show before,
Mhm.
the black smokers, which are known to be very hot, and these upwellings of like volcanic activity that have a lot of gases and the hot water that allow nutrients for uh, microbes to be able to live and have these small,
Yeah, these food chains, yeah.
yeah, food chain habitat, everything. How is this hydrothermal field different from just the black smoker
From
or
all of
one of
them,
these
yes. So
from
this
all of
is
them? Yeah.
yeah, from
Why
all
is
of
this
them, yeah.
significant?
Yeah, you just gave a really great introduction into how I have to explain this because I always have to first explain a black smoker and what that is, and then why this is different. So this vent field was accidentally discovered in 2000. It was actually the second to last day of their cruise that they were just kind of scanning this. Actually, this is a great picture to show right now too. They were scanning this transform fault wall and kind of going up and down and moving a camera. And then it was spotted out of a side camera, these like ghostly white, kind of greenish towers. And the arrow you see in the top corner there, it's pointing to where this is located in the Atlantic Ocean. So this is basically a plate where the plates are splitting apart. The plates
Okay.
in that spot are pulling apart very, very, very slowly. And so it's allowing the mantle to rise up. So that map showing all those red spots, that's, like a mountain under the water, but that's mantle rock coming up
So
from
hotter, it's the plates are pulling apart, and so you have the rising of
of the mantle.
the
Yes.
mantle up
But
and pushing the crust upwards.
it is, yes. And this is
Yeah.
ultra, ultra slow spreading. I mean, I think it's like 1 centimeter a year. Like it's super ultra slow spreading. And so it
That's
doesn't
slower than
allow
like San Andreas fault movement and like
yeah, right.
yeah.
Exactly. And so what is happening is not heat from the bottom of the earth, the magma, the volcanic activity, which is what the black smokers are getting their energy from.
Uh,
This
yeah.
is actually the mineral olivine is in the mantle, and that has an exothermic reaction with the salt water. So it is when the salt water gets into that mantle rock, when it gets down into the cracks, it is having a chemical reaction, and
Like
it is
baking,
creating
like
serpentinite.
serpentinite. Okay.
Yes. So
I,
serpentinite is I tell people think of like the crusty buildup of calcium on your faucet. It's not a great
okay.
analogy, but it's the best way people can think about it.
Yep.
Basically these big ghostly spires are the serpentinite. It's the leftover from this
Oop.
chemical reaction happening, but they're actually producing heat from the exothermic reaction. So there's heat coming off of these big spires, and you can see it. And that was one of the things that really jumped out at the researchers. They saw this like shimmery water coming off of these spires, and they were like, it's so funny to read the original NSF blog post to it too. Yeah, this video you'll see in the second shot, you'll see that water shimmering off of it. So
Shimmering.
that's all the heat coming off of it. It's not cool, but it's not as hot as a black smoker. So it actually allows microbial life to really thrive, because most
It's
black
not
smokers,
extremophiles.
yes,
Right.
yeah,
Or,
no, exactly. And so it's, even at the black smoker sites, if anything gets too close to those vent fields, they're going to die. It's way, way, way too hot. Um, but this is like much cozier for microbial life. So there's sort of like almost like a snotty film on these white vents, and people call them white smokers, which is a misnomer because they're not really smoking. But yeah, the idea is that this is a cozy place that has all the right chemical soup, if you will, that perhaps life got its foothold here. And because it is driven by a chemical process, it could very likely happen on these icy moons of Jupiter and Saturn, because this kind of reaction doesn't
yeah.
need a molten core for it to produce this heat and energy. It's doing it from a chemical reaction, not from the core of something. So a lot of the NASA missions now are specifically looking for this kind of potential of these vent fields being out there. So like the Clipper mission, which is set to launch this year, is going to try to fly through the plumes of Europa that's like spitting water out into space, and it's going to try to fly through some of this water to try to catch water samples as it passes through. And so hopefully on my expedition, we took water samples. Hopefully we can use that as an analysis to, compare and see what we might be able to find. So yeah, I shared a bunch of my videos. This is the moon pool of the ship. So I sailed on a ship for 2 months called the TWIS Resolution. It's an ocean core drilling research ship that goes all around the world. And it is, each expedition is very specific to whatever mission the ship is going on. So that was a bad way of explaining it. So like previously beforehand, it went to like the caldera in Santorini, and that was very like volcano research, right? So my expedition was very astrobiology, looking at this part of the world and taking core sampling.
How many times did they head out, or have researchers, do you know, have they been out to the lost city? Is this since was the early 2000s when it was discovered?
Yeah, so the first cruise accidentally discovered it in 2000. Then the NSF funded a huge follow-up to that it's either 2005 or 2008. I can't remember off the top of my head. But so the NSF then funded a very big expedition to go back out. Since then, I know that some universities have funded to have some work done there, and the TWIS Resolution has been back there twice to my knowledge, including my expedition. The trick about going there is that it's very remote. It is smack dab in the middle of the Atlantic Ocean. You are also pretty far away from any shipping lines. I mean, when I was out there, we saw one ship pass by, maybe two. So you have to be able to have something that can sustain itself for at least a couple of weeks, because it takes a while to get out there. And so that video you saw of the moon pool, that hole in the bottom of our ship is always open. And that makes it possible to do what we do, but it also makes the ship move really slow. So I don't know like what the speed is to get there for a normal ship, but it took us like almost 4 days because we have to go very slow because of that hole. But anyway, my point being, it takes you a few days to get out there, and then you want to be able to do at least 2 weeks of work, right? It would be a complete waste to travel all that way and have to turn right around. So, I don't know how many times it's been visited, but yeah.
Yeah, I just saw that it's been, they're not refunding it, and the TWIS Resolution missions aren't happening anymore. I'm sure there will be other missions to this area, and the TWIS will probably be replaced by other research vessels and research missions. But what was it like? I mean, the moon pool image with your commentary about this hole that's in the middle of the research vessel that you're on. It's a large hole, and you watch the waves coming in and going out, and I like sailing. But that was watching it. It's either mesmerizing, or it could really cause some seasickness, some terror. These are big waves. Like you're in the middle of the Atlantic Ocean. This is not near anything. So what was it like to be on the ship and to be exploring in that way?
Yeah, no, well, the first thing before I even left, everyone asked me, do you get seasick? Do you get seasick? I'm like, well, I scuba dive a lot and I go on boats and I never get sick on those boats. So I don't know. I do get seasick. So for the record, I do get slightly seasick, not very bad, but a little bit. So that was, kind of the first big experience was that movement heading out. It was just really weird. Like, I don't know, just that movement. And then you kind of realize like, like, what did I get myself into? Because you're heading out for 2 months. And
I better get used to this, get my sea legs.
Yeah. But the nice thing about a ship like that is to be able to send this pipe miles down into the ocean floor. The ship has to be very, very stable. So once you get where you're drilling, there are 12 giant thrusters on the bottom of the ship, and it will hold the ship in place. And then the captain will turn the ship to face the wind and the waves. So once you get where you need to go, it's not too bad. Some people are also very, very freaked out by the, like, not being able to see land for 2 months. That didn't bother me, but I know that I've heard stories from other people that it has, like, I've heard even stories of other expeditions where people had to take the ship back to port because people were freaking out so much.
Wow.
And luckily we never had any issues to that magnitude on my expedition. It's amazing. Ultimately, it's really amazing because the ship runs 24/7, and then they have a paid staff that cooks all your meals. You can have your laundry done every single day. There's a gym. I mean, it's kind of like cruising for science.
That does not sound like a terrible experience.
No, not at all. I miss it so much. um, but I miss it, but I don't, because there are so many moments where, like, on that expedition, it could not have gotten done sooner. You know, you just are just ready to be done. You're just ready to get
Yeah.
off. You're ready to be done. And
But
then
you
once
can't.
you're back, yeah, but then you can't. Exactly. So they make it as comfortable for you as possible, for
Yeah.
sure, which I really, really appreciated. But it's not for the faint of heart. And I've done an analog astronaut mission before this, where you get
Yeah.
isolated for a couple of weeks, and I would say that was harder than the ship in many ways. But it's not easy to be completely separated. You know, you can still call home, but
Yeah.
there's definitely some mental strength you need to have to be able to go out for 2 months.
So you've scuba dived. You do scuba dive. You've been on this 2-month long journey, and you also you let me know in the notes that you have climbed up to the base camp on Mount Everest.
Yeah.
So it sounds like you are not averse to stressful or risky
Torturing myself, yes.
experiences.
I wouldn't say that I've always done everything with grace, but yes, I have stuck myself in some positions where I am like, I don't know if this was the best choice, but here we are. So, yeah, I definitely am okay with very extreme environments, in the sense of being okay with the level of danger that can sometimes come
Mhm.
with those things. I am actually working on a project now with my PhD that does research up in the Arctic. And I just got back from training for that very recently, and listening to everybody else's stories about work in the field. I'm like, this might actually be the most intense thing that I'm going to be getting myself into if I do get to go out to the field, because um, even the equipment that they'll leave for just short amount of time, the bears will get to them. And so there's actually it's a very dangerous bear area to go up there. And
It's
I'm like,
very dangerous. Yes.
yeah, so it's fun. Yeah. Yeah. So that's my PI, Dr. Pash. And yeah, we're working on basically mapping, and we're using AI actually to help understand how the thawing of permafrost in that area is affecting the infrastructure and the roads, and then hopefully using
Mhm.
AI to predict, if things continue on the track that it's going, how the roads will continue to break down, and
Right.
how what we can do about it.
Yeah, are they going to remain or not? What's going to
Yeah, I
where are the sinkholes gonna form? How are they gonna
exactly.
break down? Yeah.
Yeah. Yeah.
S
And there's also like some biological risks to all the permafrost going away so quickly up there too, because there were a lot of mass graves that were dug from like
Oh,
flu epidemics and stuff, and those suddenly are getting exposed very quickly. And so there's even some looking into like what are the implications to health for that happening. So it's a really cool project to be on, just a completely different world than kind of what I was doing on the ship in some ways, because it's Arctic.
yeah.
But
I mean, Mount Everest, we know, it can get extreme, very cold the further up in altitude you go. There also are, the deglaciation that's occurring as our atmosphere is warming is unearthing corpses and excrement.
yeah,
People, old food stores, old campsites, all sorts of things on mountains all around the world, not just Mount Everest, but um, I had considered the permafrost defrosting in terms of just natural microbes that are just in the soil or just are old and
yeah,
maybe get refreshed and come back to life because why not? They're just a
Right.
bunch of
And
DNA or RNA and they're
they're
fine. Like,
doing what
whatever.
they're doing.
Yeah,
They're
I just
very,
get to sleep for a while. It's great.
they're very smelly too. I got the chance to like, I know it sounds very weird, but like thawed permafrost liquid. They're doing lots of studies on it, and it's actually quite odoriferous because it's very old. And I was at the Krell lab in New Hampshire, which is the cold regions research lab, and they have a bunch of different samples and stuff like that, and really cool science happening there. So anyway, yeah.
It's like the equivalent of geologists licking rocks. It's
Yes.
permafrost
Yep.
scientists sniffing the
Sniffing the thawed permafrost, yeah, or paleontologists. I don't know if you know this, but if you can tell something's a fossil if you stick it to your tongue and it sticks to your tongue just slightly because the little porous holes of the fossil will stick. So right, all out there.
This is not. We do not want to tell people to go out and lick things in nature without knowing what you're doing.
Yes,
But
don't do this in the chemistry labs, but maybe in nature. I don't know. Take your chances.
go for it. Yeah, lick a rock, lick a fossil.
Lick the rocks. I don't know.
Sniff some permafrost. It's great.
Yeah, why
we'll see
not?
if you get the flu. Yeah, how does that work out for you?
But yeah, no, it's been an amazing ride, and so that's my current kind of thing. But my big thing from the ship is this Lost City field, and yes, like you mentioned, the book and the planetarium show, and working on that is kind of ongoing on top of all this.
Sounds like, yeah, you're doing a lot of communicating, but I really would like to know the graduate work. So this Arctic work that you're doing, it's the research and understanding what's happening there, but you are really engaged in communications. So
Yep.
is your role in this going to be helping to communicate what the findings are, or what is your goal?
Yeah, what is my role? Right.
Yeah,
And that's
happening?
interesting. And fun part for my job is there's two sides to that coin. One side is I do need to understand what's happening so I can communicate it properly. But then I also need to be a good communicator to a general audience because a lot of these projects that I work on have public funding, and it is the responsibility of these projects to communicate to the public what is happening because it is your dollars that are being spent on whatever they're doing. And there's a huge challenge in that because I could go up to you and say like, oh, I was at the Lost City hydrothermal field where serpentinization is happening and that's causing hydrocarbons to form and blah, blah, blah. And someone would be like,
What?
nothing.
Why does that matter?
Yeah. And it doesn't matter to them either. And that's the big challenge is, and I challenge the people that I work with because sometimes I have to interview people and ask them to explain their jobs to me. And I'm like, okay, but you need to tell me now, why does that matter to Sarah in Northern Illinois? The Arctic is so far away and most people here don't care. They don't know and they don't care and it doesn't affect their life. How does it affect their lives? And that pushes them to think about it more. And then it helps me do my job. But that's really,
but it's a great role to be the person who's pushing the scientists to really get at like the nut of why it's important. Not just I'm a scientist and I'm curious about this, but like why is it going to matter to other people?
Exactly. Why is it going to matter? And then it also brings them out of their, I call it the curse of knowledge bubble, because they're so ingrained in what they're doing, they kind of forget that like other people have absolutely no idea what you're talking about. And so, I try to always find equivalents that most people can understand. So like in my current project, we're developing this platform that I would call kind of like a super sciencey Google Earth. Like that is sort of the format when you launch into the platform. You can see the globe, you can see, Alaska, and then you can click on the map and you can go see LiDAR data. And so again, these are things that like LiDAR, I don't assume most people would know what LiDAR means versus radar. And so I like that. I like that part of my job, but someone who works in LiDAR would be like, well, it's LiDAR. Duh. Like you should just know that, so I like breaking it down. And that's what I love doing as a science communicator is communicating science and then getting people really excited about it. Like there's a vent field in the middle of the Atlantic Ocean, and it's the only one we have ever found on Earth so far that works this way.
And it was found on accident.
And it was found on because of a broken
Good.
instrument too.
That's
They
great.
yeah, and I think that it's just so cool and nobody knows of this thing. And so if I can get somebody excited, then I've done my job. Like that's literally I've done my job. If you walk away and you're like, I like this weird vent field in the Atlantic Ocean, I'm like, yep.
I mean, the Lost City, that name is, I
Oh
mean,
yeah.
it's reminiscent of something out of, either Indiana Jones or something that's lost in the Amazon, some culture like, Atlantis or, some like,
Yeah. The best
'cause I
part is
don't
no one remembers who came up with the name too. It's so funny. You're like, "Who came up with this?" And they're like, "I don't know."
that I mean, that would be like, that in itself is
Right?
it's a great
And
story.
and that's what we're doing with this book project. We're trying to share the original stories and share the importance of the research of that area, but put the human story into it because that is what people resonate with and what
Yeah.
makes these places important and what matters. And so that's what we're doing with that. And then what I'm hoping with this planetarium show then is to have a very visual, high impact way of sharing this kind of science because what person doesn't just have their mind blown in a planetarium? I mean,
And I love planetarium shows, and to be able to go and experience a planetarium show that's going underwater and then potentially
right?
connects you to
Space,
Europa and Enceladus, like
right? I
fly,
know.
the visualizations that can fly you from, these miles under the ocean
Yep.
to thousands, millions of miles away from our
Yeah.
own planet. This is great.
Yeah.
I'm
Yeah.
looking forward to the day that I can like go to OMSI here in Portland and say, I I know who did
Yeah,
that.
I love that. Yeah. Well, I'm super excited about that project, and I'm really excited to bring it to my community because my community definitely needs all the help it can get with like supplemental STEM education initiatives. So that is a huge part of what I do. As you can see, my name is Space Case Sarah, so that's who I am as a science communicator in my community. But I yeah, I try to do as much outreach within my own community as possible. And it actually started with sidewalk astronomy. And so,
Love it.
again, going back to
Mhm.
if I can just wow someone just a tiny little bit, right? Show them the moon. And they're like, whoa.
I've never
I-
seen it like that.
Yeah, I've done my job. I wish I could do that every day. Just wowing people with that and kids, especially, if I can just give them that little bite and hook them because it's so unfortunate to me that so many children especially in our community the reading levels are shockingly low and so the teachers
Mhm.
are just trying to get kids to read. They're just trying to get them up to par, so STEM falls pretty far down in the pile of education. And so then their first exposure are like textbooks, which are so bland, and so they have to cram as much as they can into these textbooks. It's it's almost criminal to me. So if I can give
Next
them,
thing,
yeah,
comic books
yeah,
or, adventure books that are,
something,
they
yeah,
something
something
that gets
sparks
the
your
kids into it.
imagination. Yeah, exactly. Then I've done my job, and so yeah, it's been it's a wild journey, but the thread of it all has been communicating science.
Do you think that you will continue in the space-based direction or, kind of as that through line of
Yeah.
biology, chemistry here on Earth and where that leads into the universe?
Yeah, I definitely want to continue the thread, and I actively look for ways to see how what I'm doing, even if it might not seem like it's super linked, um, connect it to astrobiology because again, to me, astrobiology is just it's all the sciences, and I like to always tell people Earth is a part of space. Like we are on spaceship Earth, and it is no different here than it
Mm.
is out there, and so we're not as separated as we kind of feel that we are, that it's Earth and space, like, we're all a part of
We're
this
part of it.
big thing, right?
Yeah.
And, So like my work in the Arctic, I can already see sort of connecting points to this extreme cold weather, because that's a huge part of what we're also looking at is it's very hard to keep drones and batteries going in extreme cold. And those are challenges that NASA is going to face sending things to these icy moons. It's the same issues.
If you can keep an electric vehicle running in
Right.
sub-zero temperatures, then maybe you can keep a robot drone on
Right. Right.
on
Well,
your robot.
and then kind of linking back to some of the science stories
Yeah,
that we were talking about earlier, but right, there are microbes that have evolved to live in these extreme cold environments. And
yeah,
so there are scientists looking at the biology of how that works and how can we implement that biology into our technology to keep these things from basically malfunctioning because they freeze over.
right. Yeah, how do we keep that chemical energy producing either enough heat or just enough momentum chemically to maintain reactions
Right. Exactly.
so that
Yeah.
our technology works, so that our investigations work.
So all of it works. Yeah, it's all connected to me. I see it.
It's to you.
To
Yes,
me, I
brightest
see it.
center
I
of the
will say
universe.
that, yes, some of the scientists still have a very like, I'm a geologist. I don't do space stuff, and we're all on the same team, Team Earth. Okay.
Yeah. Team right now, team Earth, team life. Let's keep it going here on our
Yeah.
planet and figure out how to live here better while we figure out how
How it
other
all
life
works.
how it
Yeah.
all works around us, right?
Yeah.
Where do we
Exactly.
fit in the giant scheme
Exactly.
of things?
Yes. Well, that's getting very philosophical, but yes.
Yes.
Yes. I was almost going to go there with the muscles thing too. If a robot showed up on Earth from another planet and it had muscles that had like cells, would we consider that life visiting Earth?
I would.
Would you?
I mean,
Okay.
but it's a robot, but at the same
Right.
time, yes. I mean,
Right. I'm just throwing it out there. Would that be
this
would
is a
that
really great question. I love this. I'm gonna go ask my son about this later and get
like
him
would
to
that
talk about
be
it. Yeah.
I mean, there's a whole concept of if a robot in general visited, would it be
When
considered life because
Right,
it's not from
when
us?
is artificial intelligence, general artificial intelligence considered intelligent enough to be conscious, to
And
have
like
enough
visiting. Yeah.
awareness, sentience, sapience, etcetera,
Right.
versus I don't know.
Yeah.
And then
This is
there's the
also
life has to be heritable according to our definitions. There have to be mechanisms for heritability and for, reproducibility
right.
and for sustainability. You know, I don't know.
I don't know.
I
That's
don't
what also astrobiology deals with too. There's definitely a whole philosophical policy making side of it too, where say we go to Europa and we find life. What does that mean? And what does,
It
how do
it
we
it
how do we
means
handle that?
it means we sent a craft there that probably has microbes from Earth on
It's
it, and we just
yes,
we
possible.
just contaminated.
We probably contaminated it, right? But
Yeah.
there's a whole slew of ethics. That's why NASA has a category rating of different planets and like how at risk it is to pollute it with our own
Yep.
oopsies. And so, like the moon is a 0 because we know nothing could live on the moon. So even though
It's
certain
also close, and maybe we're like, we just kinda wanna go."
well, that too, but I don't remember. It was a few years back. Some spacecraft crashed, and there were a bunch of tardigrades on it, and that is like, oops. But it's not going to probably mess anything up because it's not really,
And
it's not
that's the
great.
question. The
But
people would love to think that those tardigrades are just having a party on the moon, but
I think they
it's
are. But
yeah,
I
it's
don't
a tardy party.
know. Someone needs to make that into a shirt now.
I think that would be great. I don't know, anybody out there and who's in our audience, take it and run with it. Sarah, I don't wanna keep you too late because I know, it is a late night for you where you are, and I'm really enjoying talking with you about your work and the things that you've been involved in. Is there anything else that you wanna just touch on before
No.
we head out for the night?
I don't think so, but thank you so much for having me. And I know I feel like there's so much that I feel like I just skim the surface of every little topic, and then I'm like, but wait, there's so much more. But you can definitely find my work and me online as spacecasesarah.com. And I'm on all the, as people would say, time wasting platforms. So
I'm sure
I
you're not wasting any time there.
all
You're
the
just
time wasting
it's all the work. Yeah.
But I like to share pictures and share memories still of especially the ship, because I haven't even, it hasn't even been a year since I was on it, so it's still pretty fresh. Like even some of the people I sailed with, they work full time year round on the ship, but they go 2 months on, 2 months off. So like that crew is back on the ship, so it's still like very like like I'll have these moments where I'm like, I just need to share a video because I miss it so much. But
I
yes,
need to
Eric,
go back to the seasickness.
yeah, no. I figured out kind of my trick for the seasickness. It's definitely, that's all an inner ear thing. So if you're working on a laptop and your world is moving around you, but you're looking at something stable, your brain is like, "Rah!" Like, I do not understand why I feel like we are moving. So it basically was like, I cannot work today. I can't, I would just have to go outside, and I figured it out. But
Find the horizon so that
yes,
you get your brain and your inner ear and
definitely
visual
the
system and
horizon.
the
Yes, that was a huge thing.
yes, make it all work together.
Yeah.
But
But
it's
yeah,
like car sickness. That's why you have people sit in the front seat of the car or be the driver so that they have like the proprioception and the vision that works with the inner ear stuff to make it all work together
yeah.
if you have car sickness.
Yeah. Some people were
Anyway.
really bad. And we also had a hospital on board with a doctor that could give you some mild drugs and then really, really heavy duty drugs if you really struggled. So,
There's only so much that Dramamine can do after a while. Yeah.
yeah. Billy, who is one of my co-author on the book that I'm working on, he basically was like, yep, you're not going to see me for 48 hours because I am going to be laying down. And I'm like, he's like, I don't know how I ended up in this line of work, because he specializes in studying the lost city. So he's always going out in the ocean. It's like, ooh, you picked a bad thing to focus your research
It's
on.
like, just give me a minute. 48 hours or so, then I'll be okay for a while. I might have to go back down again, but we're good.
Right.
And now I'm used to it. It's just the way it is.
Right.
Eric Knapp does live in Alaska. He's saying the most important thing in going to the Arctic. Clean.
Eric, where do you live in Alaska? But anyway, yeah, no, clean, dry socks. I would agree.
Just about anywhere.
I've been reading a book called The Comfort Crisis, and it's the author, he goes up in Alaska and does like a month-long hunting expedition kind of thing like that. And yes, he gives lots of suggestions of like boots and gear and stuff. And I'm like taking notes like, okay, I should probably remember that. I am such a wimp for the cold too. I would much rather be somewhere warm and diving, but here we are. So, and you live.
I wouldn't be diving. I just like warm places. The cold, I don't know.
Try diving. It's so fun.
I don't know. I've snorkeled. That was about as much as I could do.
Aww. I've been diving.
I like videos. I like looking at what other people do. It makes me very happy.
Right, fair enough.
Sarah, thank you so much for joining us tonight. As you said, your website is spacecasesarah.com. Sarah Treadwell. I expect that we are going to be seeing some really incredible stuff from you in the future, and I can't wait. And I hope that we stay in touch and are able to find out more about your adventures and your communications work, because it's good stuff.
Yeah, it is. It's just pretty exciting stuff. Sometimes I'm not even like sure that this is my own life. I'm like, who? Like, you what? Me? Yeah.
It's adventure, Sarah. There we are. Yes, it's good.
Yeah.
So everyone out there, Sarah, once again, short notice, and I appreciate you being here tonight so much. And it's just been a wonderful conversation. And I hope everyone else who listened in and was here during the show appreciates it. I hope everyone who catches it as a podcast also enjoys the show. Thank you for being here. Thank you for listening, everyone. And I do have some shout outs for people who help the show on a weekly basis. Fada, thank you for your work on show notes and the social media. Gord, Arnlor, others, thank you for helping out in the chat rooms and keeping everything happy. Our Discords, our YouTube, Twitch, and Facebook chat rooms. Thank you, everybody who was here live and chatting in the chat room. Identity 4, thank you for recording the show. And Rachel, thank you for your amazing assistance in editing the show. And of course, I must now thank our Patreon sponsors. Thank you to Aaron Anathema, Arthur Kepler, Craig Potts, Mary Gertz, Teresa Smith, Richard Badge, Bob Coles, Kent Northcote, Rick Glovman, George Corus, Pierre Belazarb, John Ratnaswamy, Carl Kornfeld, Chris Wozniak, Vegard Shefstad, Donathan Stiles, a.k.a. John Stylo, Ali Coffin, Reagan Shubru, Sarah Forfar, Don Mundus, P.I.G., Stephen Alberon, Daryl Myjax, Andrew Swanson at Fredis104, Sky Luke, Paul Runovich, Kevin Reardon, Noodles, Jack, Brian Carrington, David E. Youngblood, Sean Clarence Lamb, John McKee, Greg Riley, Mark Hessenblow, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Richard, Brendan Minish, Johnny Ridley, Remy Day, G. Burton Lattimore, Flying Out, Christopher Dreyer, Greg Briggs, John Atwood, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Rick Ramus, Philip Shane, Kurt Larson, Craig Landon, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, Lon Makes, EO, Adam Mishkan, Kevin Parachan, Aaron Luthen, Bob Calder, Marjorie, Paul D. Disney, David Simmerly, and Patrick Pecoraro, and Tony Steele. Thank you for all of your support on Patreon.
Did you breathe?
Barely, yeah. I'm trying to be like the old school, what is it, the little matchbot machines. Anyway, micro machines, the little cars. Oh, if anyone else is interested in supporting us on Patreon, please head over to TWIS.org, where you can find a link to our Patreon page. Next week on the show, Justin should be back with us, I think. I don't know. Don't miss it. You don't know what's going to happen. I don't either. We'll be back. I will for sure be back broadcasting Wednesday, 8 p.m. Pacific time on Twitch, YouTube, and Facebook. And if you want to listen to us as a podcast, of course, just look for This Week in Science, all places that podcasts are found. If you enjoyed the show, get your friends to subscribe too. And if you have any need for more information or links or any of the things that Sarah and I talked about tonight, again, our show notes are on twis.org. And there is also, you can put your email in and subscribe to our newsletter if you want to. That's all there at our website. You can email us as well. My email is Kirsten at thisweekinscience.com. Justin is twistminion@gmail.com. And Blair is blairbaz at twist.org. Don't forget to put TWIS in the subject line so your email doesn't get spam filtered into the lost city hydrothermal vents where, it's lost. They kind of found it.
I will find it.
I
I know that place now. I have all the maps. So you tell me, I'll find it.
I'll tell you. Oh my goodness. You can find us on social media. We're still out there at TWIS Science is the main account. My account is at Dr. Kiki. Justin is at Jacksonfly. Blair is Blair's Menagerie. And Sarah, what is your usual socials?
Oh, I'm Space Case Sarah everywhere. Every single time wasting platform, you type Space Case Sarah, you shall find me as such.
Wonderful. If anyone also wants to request a topic or an interview that we might do, someone like Sarah to guest on the show, let us know. We love your feedback. And we will be back. TWIS will be back here next week. And we hope that you will join us again for more great science news. And of course, if you did learn anything on the show tonight, remember, it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.